This is a production of Cornell University.
Thank you very much for the introduction, Dr. Krasovich. And I want to say good afternoon to everyone. And I want to thank the Plant Breeding and Genetics Seminar Committee and the chairman for this opportunity to present on the activities of the Innovation Lab for Crop Improvement. Here is a snippet of what I'm going to talk about today. I'm going to give us a brief background of myself, and I'll go ahead and introduce you all to the Innovation Lab for Crop Improvement. And I'm going to round up by talking about our centers of innovation, which I believe is the heartbeat of ELC. So for those of you who do not know me, I am originally from Nigeria, just like I was introduced by Dr. Krasovich. And my first introduction to plant science research was through IIDA. So that was where I received inspiration actually to pursue a career in plant research. Prior to that encounter, my family routinely processed cassava to make gari ourselves, just for personal consumption, not to take it to the market or anything like that. So the men would harvest cassava and the women were tasked to turn the cassava into gari. And we would peel and process into gari. And that was short-lived because of the death in the family, as the breadwinner died. And then life, especially feeding for, I was just talking about a 7-year-old, feeding became very difficult, and a child, what does a child know rather than to eat breakfast, lunch, and dinner? And that was the point in my life where I witnessed poverty, firsthand and experienced food insecurity. And of course, all the while I desired to find a solution to these problems because children shouldn't have to worry about, where their next meal is going to come from. And not knowing, I would end up working in crop improvement research and cassava for that matter. So after this encounter, I was privileged to serve again in IT. That I was able to do for a year. And I worked with IT Biotech at that time. And it's now called the IT Bioscience Center. After which I was inspired to continue in plant research to obtain a master's in crop protection and pursued my PhD in Germany. I did the molecular biology work at the Max Planck Institute for Plant Breeding Research, and I had the cassava transformation work at CIAD. And the theme was proof of concept that genes from Arabidopsis thaliana can regulate flowering in cassava. Because as we all know, cassava is recalcitrant to flowering, and flowers are very important for breeders. So this research was so interesting that we had flowers, we have cassava in vitro plant flowering, cassava flowering in vitro. So we were able to prove that the FT overexpression increases the prolificacy of flowering production and extends the longevity of flower development. And I also characterized the components of the circadian clock upstream of the FT in cassava. So after I obtained my PhD from the University of Cologne, I tried to secure a position in the African continent. And that was kind of a struggle. But by divine providence, I found my way to the US and I came to Texas where I began my immigration journey. And that is where I studied the mechanisms by which legume plants, in this case, Magicago truncatula, control symbiotic nitrogen fixation, acquiring nitrogen without the use of energetically and monetarily expensive and polluting synthetic nitrogen fertilizers. So typically we would search for mutant lines in Oral Roberts Noble Foundation is close by. And then the mutant we found showed nitrogen deficiency symptoms rather. The mutant fails to form pink nitrogen fixing nodules that occur in the wild type symbiosis, but instead It develops small bomb-like nodules on its root that are blocked at an early stage of development and has a lateral root defect. The functional NIP-LAD gene was cloned and shown to encode a protein in the broad substrate nitrate reductase transporter family, and the NIP-LAD lateral root deficient transporter is essential for nodulation and appropriate root architecture. Next slide. So here is some of the publications, and after that experience, I came to Cornell, where I worked with Team Setter, and I continued, came back to working with cassava after that detour on overexpressing Rhodopsis ft that elicits extremely early flowering, indicating that cassava has the necessary signaling components to interact with and respond to the FT gene product. So my work was to advance our understanding of the molecular and physiological basis of flowering in cassava under different environmental conditions, where I investigated the expression of cassava FT-like genes. We characterized the phosphatidyl ethanol amine binding protein family of genes. So it happens that in cassava we have 10 of them, and we identified like a putative FT-like homolog. FT-like is like in Arabidopsis, it's the rate limiting gene that is responsible for switching from vegetative to reproductive production in plants. And we compare the expression of these homologs in various tissues, and we elucidated the expression of these homologs in various experimentally controlled photoperiod and temperature environments in relation to their effects on cassava regulation and other important roles in cassava. So we were able to advance the potential for applications in breeding program. Ultimately, we found out that in some of the assertions that we used, the cassava is a long day. It flowers early under a long day photoperiod, and if you put them in short, they flower late. So when I rounded up my research, my postdoctoral experience, I was serving on the board of the Cornell BEST program. BEST used to be called Broadening Experiences in Scientific Training, and it's now known as Careers Beyond Academia. It's a platform where PhD students and postdocs can find a fulfilling career outside of academia. It's more like you're test driving, different career options. So even those who pursue academic careers will need to develop skills beyond the technical or research skills they're developing in their current programs. So BEST provides opportunities for increasing awareness of the broad range of careers that exists. And examples are like in policy making, governance, risk and compliance, industry, entrepreneurship and management, communication, and research administration. So when I now found myself in the Office of the Vice Provost for Research and the Office of the Vice Provost for Academic Integration, it was an excitingly different environment for me, where I learned more about research administration, Cornell leadership, compliance issues, the Office of the Sponsored Programs, RIA. I worked on special projects. I worked with our research division, communications, and CTL. Later, I administered a startup incubator for commercialization of Cornell Technology Ventures in Engineering, Digital and Physical Sciences, called Praxis Center, just like Dr. Kresovich mentioned, where I managed the diverse administrative functions of 6 independent venture teams with up to 50 individual client team members resident in the center. For those of us who don't know Praxis Center, it's like a sister center to the Kevin McGovern Center, which is a startup incubator for commercialization of Cornell Life Sciences technology. So at that point, when this current position came up, I became excited and ready to take on this challenge, inspired really by the potential impact that this work that the Innovation Lab is doing could have not only on the African continent, but also on millions of people around the globe. So I thought that this is a perfect opportunity for me to fulfill like that desire. And I found myself having a career trajectory that starts with research and continues with a large ag research grant management. So that brings me to ILSI. Excuse me. So talking about Innovation Lab for Crop Improvement, we cannot talk about Innovation Lab for Crop Improvement without talking about the Feed the Future and the global food security. So the Feed the Future is a US government initiative to sustainably reduce global hunger, malnutrition, and poverty through agriculture-led economic growth to strengthen resilience and improve human nutrition. This is led by the USAID. It was a Global Food Security Act, and then it's implemented by the Global Food Security Strategy. And these are the US partners that are involved, the US government agencies and departments that are involved in this research. And the Feed the Future has 12 target countries. So the implementing partners are important, and these implementing partners are research funding organizations, they're innovators, and development partners. I would say that there are about 21 Feed the Future Innovation Labs that ranges on topics about animal science, research, to policy, food policy, and to plant research. And the ILSI was previously, there are innovation labs that specifically deal or work on specific crops. But the ILSI is working on a broad range of crop with a global impact. And this was awarded, it was a competition, and Cornell was awarded. We're excited that Cornell was awarded. It's a 5-year effective last year, and the award amount is 25 million, and it's to develop a crop improvement portfolio for the NARIs. So what is our mission? Our mission is to partner with national programs to help them identify, develop, pilot, and transfer appropriate tools, technologies, and methods to equip them so they can deliver increased genetic gain for better seeds into the market for smallholder farmers. So we ultimately reduce malnutrition and hunger. Our approach. So historically, donor organizations will provide funding for research, but they drive the goals and priorities for research. But in ILSI, we have this approach of bottoms up approach, where the ILSI, the NARIs, they drive the goals and priorities for research. Why? Because we believe that sustainable solutions will have a long-term effect if the NARIs specifically identify their unique needs, and target innovations or design innovations to solve them. So, and we focus on providing, resources in terms of cutting edge science and collaboration and all that they need, that we believe that will help them to succeed. So what is our tactic? Our tactics include this core interdisciplinary team of scientists that focus on priority setting, of breeding interventions that will increase the responsiveness of these interventions and potentially improve uptake of new varieties. We have a trait discovery team, genomics, phenomics, breeding informatics, and institutional capacity evaluation that we believe are the core core research areas. We call them area of inquiry that are essential for success in the NARIs. And here is like a sketch that talks about the area of inquiry, deploying tools, technology, and methods through a breeding pipeline for a healthy crop improvement system. And I'll take you quickly through the management team. We have Dr. Kresovic, Harley as our associate director, and Monifa as our program assistant. And we also have the team of communications support and administrative and event personnel. Just a brief going through the area of inquiry team leads the faces behind the tactics. These are our core area of inquiry team, and we have Jeff Morris from Colorado State. We have Jesse Poland from Kansas State. We have Dil Thavaraja from Clemson University that works on phenomics, and the rest of our team are all Cornell based. We also have a cross-cutting theme. We believe that we have a cross-cutting theme lead, designated lead in Elizabeth, and we have Dee and Margaret also working together. And the cross-cutting theme is very important in that it's about increasing awareness and understanding of the importance of addressing and incorporating gender, youth, nutrition, and resilience in crop improvement. I want to highlight two things that the cross-cutting theme team have done so far. And that is training and translation. Training. Due to COVID, we were just talking about COVID-19, it has been difficult to travel in person. And with the rising numbers of infected cases, travel restrictions will probably not ease. So our cross-cutting theme, they have designed, outlined, and online, sensitization training to reach our COIs more quickly than would have been in person, so that work could continue despite the travel restrictions. And I think that this is a very great idea that they have initiated 2 trainings already in gender and youth to serve as a framework for additional 2 trainings in nutrition and resilience. And the trainings will also serve as a public tools accessible by any way, as well as templates for the other area of inquiries who may wish to pursue the same for their own area of expertise. So the other accomplishment that they have done is that we know that all the centers of innovation I'm going to talk about later, they are awarded in countries that are primarily non-English speaking, or they speak English as part of its colonial heritage. And we have stated that the objective of the ELC includes diversity and inclusion. And we have reinforced those through our strategic management and prioritization of cross-cutting themes throughout the project. So they are working to formalize access. In fact, I must say also that I just read an email that was sent to one of our COIs in Senegal in French. And I'm impressed that, we are working so much to make sure that ELC succeeds. And if that means that we want to translate, the work to remove any communication gaps, so they're working to formalize access to translation and or interpretation services to support inclusive outreach and capacity building for the cross-cutting theme, as well as the ELC team and initiative. All right, and then I'll just go through our advisory committee. Our advisory committee they are seasoned professionals with international experience in leading research and development in the private sector. And one of them is linked with the private sector in Asia. And together, we believe that our core team, area of inquiry leads, and a cross-cutting team, together with the management entity, will leverage these connections to engage efforts that connect with private sector priorities, so we can ensure technology and method advancements and transfer them to advance our NARI partners. Now it takes me to our target crops. Our target crops are root tubers and bananas, legumes, excluding soybean and peanut. We have millet and sorghum. And I would also talk about the quick wins. Who are the quick wins? The quick wins were identified as short-term projects with a moderate investment and for a long-term impact, and were identified at the time that the proposal was submitted. And we have quick win partners in Uganda. We have two in Uganda. We have one in Haiti, we have one in Nepal, and one in Senegal. So a quick win in Uganda, one of the quick wins in Uganda is working on weevil resistant sweet potato. A quick win, another quick win in Uganda is working on fast cooking iron and zinc biofortified bean using genomic selection pipelines. And our Nepal quick win is working on high protein quality lentil using non-destructive phenotyping tool. And our quick win in Senegal is working on staggering and drought resistant pearl millet using quantitative traits, using backcross nested associated mapping, excuse me. And our quick win in Haiti is working to increase the rate of genetic gain in low budget developing countries sorghum breeding program using genomic assisted selection. So I would share some of the progress he has made so far. And his product profile is sugarcane aphid resistant multipurpose sorghum. He works on proof of concept to demonstrate the possibility of enhancing genetic gains in sorghum with rapid cycling genomic selection. Excuse me. So it has several genomic cycles of sugarcane aphid resistant multipurpose sorghum with broad adaptation to tropical growth environments. Those were completed. And then it went to genomic cycle selection 3 that shows selected families with yields exceeding the yield of the phenotypic recurrent selection. And the best breeds from this genomic selection cycles and phenotypic recurrent selection cycle are currently under multi-location trials. And you can see that the genomic selection cycle 3, which is under a lot of stress, water stress and severe aphid infestation, yielded, it had increase in grain yield. And is going to continue some work to validate new varieties in participatory trials. It's going to validate ID types and evaluate new training set for more locations. And I'm going to talk about our centers of innovation, and it's I believe they are the heartbeat of the ELC. So first of all, to identify the centers of innovation, we requested for an application because our goal is to build a top-tier scalable breeding programs and co-create a toolbox package covering all the 6 areas of inquiry. And we had a goal to select 1 NARI in each region: the East African region, the West African region, and the Southeast Asia region, and the Latin America and Caribbean region. And the requirement was that they write a proposal for projects for 3 years and define clearly the product profiles for target crops the RFA will impact. It must be led by NARIs in the future countries, and they should articulate the development goal and clearly indicate how becoming a COI will increase benefits for women, youth, climate resilience, and nutrition, still on the global food security strategy 3 objectives. And the budget is a maximum of $1 million. And tentative start date was October 1st of this year. Priority was given to institutions that have linkages with US minority serving institution, the minority group of women and early career scientists, and also essential that they partner with NARIs, universities and institutions, and CGR centers. And proposals should be submitted, well, an advantage is if it's submitted by interdisciplinary groups of PIs. And they should include capacity building plans that is articulated at the individual and institutional level. And then their product profiles should address gender and youth-based constraint, and they should show potential for all these three global food security strategy objectives. So the RFA process was started by the reading questions and webinars. So for the webinar, we had about 150 registrants from 41 countries. And the letters of intent, were submitted by 104 applicants across 26 countries. And we filtered those 104 applications to about 40 using different criteria. If they followed, if you were going to research in a mandate crop or have NARI PI or the country is eligible, and other supporting documents. And 12 was shortlisted. During the co-creation phase, we merged, out of those 12 shortlisted candidates, we merged two of the projects together, and then there was like a matrix of calls with the area of inquiry and the cross-cutting theme leaders to make sure that they prepare the proposal to reflect all the priorities of ILSI in the areas of inquiry and the cross-cutting theme. And then we had 10 final proposals that was submitted, and four was granted. These four are Costa Rica 80. These are the ones that emerged, and the one from Senegal and Uganda and Malawi. We have already worked with 80 in Senegal. So it's really exciting to work or have footprints in the global spectrum. And we are also working to include work in Asia. So the centers are the Central America and Caribbean Crop Improvement Alliance, CASIA, the Center of Innovation for Crop Improvement for East and Southern Africa, CCSA, is in Malawi, with Michael Chepeda, and the East African Center of Innovation for Finger Millet and Sorghum in person of Scovia Adekini at the National Salmon Allied Resources Research Institute in Uganda. And we have Crop Improvement Innovation in West Africa. And that's in person of Guido. Oh my God. So the Central America, the CASIA, is led by Jose and Gael, and the NARI is Instituto Nacional de Innovación y Transferencia y Tecnología Agropecuaria INTA and Cascaya University, and their crops are common beans, sorghum, and sweet potato. So they have the year they're set out to work on. Their year plan is they want to validate product profiles in Costa Rica and Haiti, and they want to test uniform regional nurseries using participatory approaches and expand the sorghum rapid recurrent and genomic selection approaches both in Costa Rica and Haiti, because Gael is already working on sorghum, and also they want to incorporate physiological breeding. And with the beans, the crop, they want to begin the magic population development. And for their sweet potato, they will work on rapid recurrent selection and physiological breeding. And the second Center of Innovation I'm going to talk about is the East African Center of Innovation for Finger, Millet, and Socom. The lead country is Uganda, and we have partner countries in Karo and Tari. And excuse me. And the crops they're going to work on is sorghum and finger millet. And the plan that Scovia asked for this year is that she wants to conduct a finger millet and sorghum value chain and do like a market analysis to document the drivers for adoption of technologies. So, and then she wants to identify sources of resistance to some stresses and also nutritional quality sources. She wants to do gene and molecular market discovery for novel traits and develop and optimize tools and methods for efficient phenotyping and rapid regeneration and advancement of desirable traits, and digitize breeding programs for targeted breeding in East Africa. This work is very, very important because we know that having the right tools, technologies, and methods, they will be able to execute this. And she also wants to build like an irrigation system, because I understand that it's a semi-arid area, so there's drought. So to have more, to expand the nurseries, they need to be able to keep the plants alive. So, and they need water. So these are one of the major investments that we are going to be making. And the next crop Center of Innovation is the Crop Innovation in West Africa. CY, it's led by Guido, and the PI, co-PI is Marame, and they have affiliated countries in Burkina Faso, Inera, and Imrad in Niger. And the crops they're working on is millet, sorghum, and cowpea. The work they have set out to do in year one is to update their existing breeding product profiles to seek to understand adoption differences and improve like market accessibility for the sorghum, the millet, and cowpea. And they want to implement modern TTMs for rapid discovery and validation of traits for genetic gain and product concept development. And also wants to use genomic selection and whole genome prediction of crop breeding values using simulation and GS pipelines for optimizing the West African breeding programs. And it's going to do this using modernized phenotyping and data capture platforms of breeding pipelines, and ultimately digitize breeding activities and fair data, and that face the capacity of computational systems to find, assess, interpret, and reuse data. And now the last center is a Center of Innovation for Crop Improvement for East and Southern Africa, and is led by Michael Chipeta, who is the principal investigator, and Jessica is a co-PI in Malawi, and the partner countries are Mozambique and Tanzania, and they are located in Lilongwe University of Agriculture and Natural Resources, and they're working on cowpea. So year one plan is to conduct gender-sensitive cowpea value chain analysis, and that is more of designing like study tools and framework for comprehensive gender-sensitive cowpea value chain analysis using questionnaires and checklists, and conduct trait preference surveys. And it's going to characterize phenotypic and genomic diversity cowpea germplasm, and phenotype selected germplasm or breeding lines and collect for key traits in multiple sites. It's going to genotype some of the cowpea germplasm and we sequence cowpea founder lines and just like some nutrition variety profiling, and under developing and selecting more productive and nutritious cowpea lines, it's basically selecting like the best parental lines for generating crosses. And I included capacity development here as an important part of our work in ILSI. It's important that they develop capacity so that they can self-sustain, and they want to train on the deployment and implementation of BMS in Malawi. We want to train on gender-responsive breeding and train on genomics-enabled breeding and informatics. It's clear that they lack all these tools. So these are some of the targets, and they want to ultimately train postgraduate students and project personnel who can continue to work in these 3 countries. So if they have this capacity, institutional development, they can be self-reliant or to continue to sustain themselves. So I think that's very important part of their year 1 plans. And with that, I have come to the end of my talk, and I'm excited that we are working on these different regions, because I believe that ILSI is strategically positioned to deploy crop improvement research to the NARIs. And one of the take-homes is that NARI is our brand. We are working for the NARIs so that they can be self-reliant, they can self-sustain, they can breed their own crops, just so that they can eliminate food insecurity. And I believe that our COIs and quick win research will accelerate breeding of locally and regionally adapted crop varieties that target smallholder farmers and other seed systems related outreach, so that their work and their product can be received by the end users and families and people that need the seeds. And ultimately, of course, that's the ultimate goal, to advance and fulfill the 3 global food security strategy, which is economic growth, resilience, and human nutrition. With this, I'd like to thank our partners from Kansas State, Clemson, and our COIs. And I've come to the end of my talk. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.